So yeah, I'm gonna talk today about project I've been working on the past couple of years with mapping pH and lime requirement using remote sensing, using a combination of data from our long-term research site and some on-farm study. So I think we know that soil acidification is increasing in the Pacific Northwest, and there's a number of causes for this. There's acidifying fertilizers and removal of crop residues and applying lime in a number of forms can help improve this situation. But it's really quite expensive. It's difficult to map because in order to get a good prescription map for lime, you have to take a lot of soil cores and then have them tested, and that's time and labor intensive. So I was hoping to find a way to not completely eliminate the soil sampling, but to reduce it using some remote sensing. So first I'll discuss the two research sites that I used for the study. The first one is the Cook Agronomy Farm that's located north of Pullman. It's about 92 acres. It's mostly the tuna and naphtha soil series. I focused on the east side of the field, which has been no-till since 1998, and we conduct yearly hand harvest sampling every year at 369 points, and we've conducted deep soil cores in 3 years at 177 points. And so some of the data we have from that includes soil pH. So this provides us with a good data set to study some long-term trends in pH and how that relates to other variables. And here's a picture of the plot in question and the soil sampling sites. The other site, it was a privately owned farm near Palouse, Washington. Total farm is about 400 acres, but just because, I have, like me personally, I have to physically go out and take all the samples. We decided to reduce it to just the 96 acre portion of that farm. It's similar soil types to Cook. It's also no-till. And since I started working with the farmer, the crop rotation has been garbs and spring pea, followed by winter wheat, followed by spring barley. And then next year he'll be planting garbanzos again in the spring. And so on this farm, I took 10-centimeter soil cores, and I tested pH and modified Melick buffer pH to determine the lime requirements in the fall of 2022 and again this past fall. So I have 2 years of soil pH and buffer pH data from this site. And here's a picture of the plot. And it's basically divided into two sections. There's kind of like a little road going in between the two of them along a ridge. And the black dots are some soil sampling locations. So my strategy going into this was to use the wealth of data we have from Cook Farm to look for some patterns relating pH to other variables, including yield trends, remotely sensed variables, topographical variables, soil type, things like the presence or absence of an argillic horizon, and then fit a model to predict pH using those. And then I would use that model to select the best points to sample on the private farm. So that is the key part of this, is using a model to select the best points to sample so that you can get the most bang for your buck in terms of sampling. And I wanted to figure out what was the number of points I needed to take in the field combined with remotely sensed and other variables to get the lowest error estimates. so that we could make better lime requirement maps. And the goal came from the farmer. He said he wanted to try and get it down to less than 1 point per acre, which is pretty good. I mean, considering that if you look at other sort of sampling like this, for instance, for soil organic carbon, if you look in the literature, they recommend more like 2 to 4 samples per acre. And so there's a few strategies you can use for sampling. There's grid sampling. You just sample on a random grid, on a fixed grid. There's random sampling. You just randomly select points in the field. Stratified sampling, where you might break up the field into zones based on certain characteristics and sample evenly in those different zones. So here's the overall flow chart of this over the last couple of years. So I started with the Cook Agronomy farm data, and then I fit model to predict soil pH using other variables. And then I use that model to simulate some sampling campaigns on the private farm. And then I took the best points that I found for that and conducted a sampling in the fall of 2022. And I looked at that data. I updated the model using this new data. And then I did the same thing again for 2023, this time with the updated model. And then finally, this just a month or two ago, I made a lime recommendation based on the model that I found. So here's just a picture of the sort of pH distribution at 10 centimeters that we see at Cook from 99, 2008, and 2015. You know, the overall trend is that it's acidifying, which is not going to be a shock to probably anybody on this call. And similarly for the 20 centimeter, it's also acidifying. So then when I looked at this Cook data, I noticed, you know, there's a lot of noise. There's a lot of unexplained variability. But the main relationships I found were that the pH at 10 and 20 centimeters depths were related. You know, that's not a surprise. Sort of paradoxically, the peak NDVI was negatively correlated with pH. And so this kind of puzzled me, but it's what the data showed, you know, with basically that with higher NDVI means higher productivity. And that was associated with lower pH, but that didn't make sense to me. And I'll get to that later. And then the face, so topographical face is whether a slope in the field is north or south facing. And then I found that the pH was typically higher on south facing slopes. So those are the main relationships I found to construct this model. So using this model, I used it to estimate the pH on the private farm. And I can do this using just publicly available data. I was able to, you know, I can get NDVI that's publicly available through Landsat. And same goes for topographical information. And then I conducted simulated sampling. So I would randomly select points in the field according to certain sampling schemes until I came up with a solution that met our requirements. So just so you can get a picture of what this actually means, what this looks like in terms of different ways to sample. So there's grid sampling in the upper left-hand corner. And this is just like the name sounds, you're sampling on an even grid. This is considered to be the least efficient. You know, you really have to sample more points here to get an accurate result. Random sampling, you know, you're just randomly picking points in the field. It doesn't perform too much better than grid sampling. And then the other one is I've seen used in other soil sampling approaches, and I decided to use it for pH here. And that's k-means clustering. So what this means is I grouped the points in the field according to different variables. So the k refers to the number of groups that you have. So in this case, I basically grouped the field into 5 different types of regions. And then I sampled evenly from each of those types of regions. And I found this method using 5 groups with 15 points sampled per group, ended up with the least number of points and to achieve the same accuracy as a higher number of points with the other sampling methods. So going forward, I went with the k-means clustering approach. And so it ends up being a total of 75 points for the 2022 sampling and 72 points for the 2023 sampling. So that achieved the goal of getting down less than 1 point per acre. And so I conducted my sampling according to the strategy, and this is the data. So you can see for 2022, it can be hard to discern the pattern just looking at how it's distributed on the map here. But the main thing I noticed was near the edges of fields, we tended to see higher pH. And then similar patterns for the buffer pH. So what I did at this point was I refit the model for the final time according to this new data that I had. And I found a number of other trends that made some more sense. So there was the presence of an argillic horizon, that's the BT horizon. It meant the crop was less productive in that area, which is something you typically see. And I found that the time of peak senescence, so when is the crop senescing fastest? The earlier in the season that the crop senesces, this is something I could get through remote sensing. The earlier it senesces, that means that the pH will tend to be lower, which makes sense. The crop is more stressed, it's going to senesce earlier. Another way of thinking about it is if it's senesces later in the season, that means it's higher pH, the crop is less stressed. I also found that pH was positively correlated with NDVI, which is the peak productivity. So that makes sense. This makes more sense than the previous results. So more productive areas of the field tended to be associated with higher pH, which makes sense. So that was something I could tell through remote sensing. There were some other things I noticed for the buffer pH, that the slower that the NDVI increased over the season, that was also associated with lower pH. So what this means is that at a low pH, the crop basically grows quickly and senesces fast. And then with a higher pH, it's growing a little more slowly and senesces later. So that kind of makes sense. And then finally, I found the same result for topographical face. On south-facing slopes, we had higher pH. So just a bit of explanation here would be that higher peak NDVI is associated with healthier vegetation. And that is associated with higher pH. Earlier senescence was associated with crop stress and lower pH. On south-facing slopes, we have a higher buffer pH, which means, this is because on south-facing slopes, there's less base cation leaching, and the south-facing slopes, due to soil forming factors, tend to have less deep soils and more base saturation with depth. So these are sort of the main trends and explanations I saw. And then just to help visualize what this looks like in the field. So you can see here, I'm just plotted up what areas of the field were north and south facing. So north is kind of like the orange and south is kind of the blue. And then here's the presence of the BT horizon. Blue is areas where we do have a BT horizon, and this is according to soil survey data. So, that has some limitations on accuracy, but it's what I had. And then if I look at peak NDVI, so remember, peak NDVI is associated with productivity. And especially in 2023, you can notice that the areas with lower productivity also match up with the areas where we have a BT horizon. Okay, so that tracks. And then if we look at growth rate, peak growth rate, it's lower on the south-facing areas. This tracks with the other variables. And then finally, this variable I've called NDVI5 that's the date of senescence relative to planting date. And I've normalized it here, so it's easier to see the differences. But you could see there's some variability in this. We see more variability or more apparent variability actually in 2023, which is following a spring barley season, versus 2022, which is winter wheat. And this is because winter wheat, it has a deeper rooting depth, so it's less susceptible to changes in pH relative to spring barley, which is more sensitive. So we see some more extreme changes in the time of senescence in the spring barley. And so that I put this all together to create some first maps of buffer pH and soil pH, and use those to create line prescriptions. And I included spatial correlations. So if we look here, this is the final, maps using this data, remotely sensed data, combined with the observations to make some maps of the soil pH. But, more importantly is buffer pH in terms of estimating what the lime requirement is. And you could see it's sort of the higher pH areas, you can see how they kind of track with the south-facing slopes and with areas where there were differences in time of senescence. So in terms of the lime rate calculations, I did this hypothetical as if we were going to apply lime this coming spring. And I used the Western region's lime rate calculator to estimate what the lime rate would be. And then I used some of Carol's work on adapting this to the soils we have here to scale it to an actual lime rate. And then I had to scale it again because those were designed for 15 centimeters and I scaled it so it was for 10 centimeters. And for this exercise, I assumed it was 70% effective calcium carbonate equivalent sugar beet lime. Although, obviously this number will vary depending on what kind of lime we're using. And I use the predicted buffer pH to estimate what the lime requirement would be. And then I divided it into zones. So, because there's some limitation on the accuracy of any kind of lime application device, you're not going to be able to precisely gauge the rate. So here's a map of the lime requirement in tons per acre. It actually seems fairly uniform. It's more illustrative to look at it in terms of zones, though. So I broke it up into 5 zones. And really, we could combine this into 2 zones, a high zone and a low zone. So I would say this would probably lend itself well to having a one zone, which is about 0.9 tons per acre of lime, which is kind of the orange area. And then another zone, which is a 0.7 tons per acre of lime, which is the blue area. So those are the lime rate zone maps I came up with using this method. So what are the next steps? Initially the plans were to apply lime according to this prescription map. However, lime is really expensive, so that's not going to happen. So we're going to sample again next fall to get some additional data, keep updating the model. We might try doing some smaller tests, like testing out the prescription map on a smaller portion of the field. And another thing we would like to test is something that we've been testing at Cook Agronomy Farm on a small plot, which is subsurface applied liquid lime. So there's a chance we might apply this on this private farm and test it out there. So that's about it. I guess we might have some time for some questions.